This video is lecture video 3. This is going to cover the cell and cells and how they function. There are some optional videos if you want to check them out, link through Canvas and through the PowerPoint. These are the learning objectives that we'll cover, and we'll focus on a couple of them that are a little more complex, like membrane proteins and ATP when we talk about cells. So cells are sort of the most simple living structure we have in our body, or the smallest. They're not so simple. The smallest living structure we have in our body that's actually alive. But cells in our body come specialized with not only their structure but their function. So we might have muscle cells that contract, keratinocytes to cover the outside of your skin, erythrocytes to transport oxygen. We'll focus a little more on a generic cell and some of the traits that they share. And as we go through the semester, we're going to cover lots and lots of different cells in the body and how they are specialized and how they function. So we're going to start with the cell membrane, its structure and its function. So here's a typical textbook image of a cell. It's been cut open, and what they cut open was this barrier between the outside and the inside of the cell called the cell membrane. The cell membrane is important because it's really how the cell interfaces with the environment and also controls what gets in and out of the cell. Our cells float around in extracellular fluid. The basic building block of our cell membrane are called phospholipids. Each phospholipid has a little head and a little tail, and they usually will spontaneously set up a bilayer, meaning there's two rows of these phospholipids. Phospholipids have a glycerol and fatty acids, and then you stick a little phosphate on there and you make a phospholipid. It turns out the phosphate glycerol head likes water and is hydrophilic and attracted to water, and the tails, the fatty acids, are hydrophobic, and this is how this phospholipid bilayer sort of spontaneously sets up and creates our cell barrier. Inside our cell, if we want to create compartments like the nucleus or endoplasmic reticulum or mitochondria, we'll use these same phospholipid bilayers. The same sort of thing will create a membrane inside your cells. So the phospholipid bilayer is a barrier. Some things can get through. There's other molecules in there like cholesterol, which helps its structure. And then what we're going to focus on, one of the more important things that our cells stick in their membrane or attach to the outside and inside of our membrane, are called membrane proteins. So a lot of things can't get into your cell unless they actually go through a membrane protein. So for example, sodium ions and glucose. Sometimes our cells are going to want to move things in and out of our cells. And we also can put things like carbohydrates and other things stuck onto our membrane as well, but we're going to focus on proteins. Some of these proteins are anchoring or adhesion proteins so that cells can hold on to each other. And so for learning objective 3, we're going to look at some of the most important membrane proteins and some examples of them that we're going to see throughout the course. So proteins, remember, are functional molecules, so membrane proteins are going to have important functions, everything from binding hormones to drugs to preventing cancer. So here in our diagram we have the inside of our cell and the outside of our cell separated by a phospholipid bilayer. We stuck on some membrane proteins and we're going to look at examples of what they do. There are sort of 5 things that I decided membrane proteins do. You might want to pause it and write down some of this list in your workbook, and we're going to go through each one examples. Starting with channel proteins or ion channels. So ion channels are specific proteins in our membrane that are going to allow ions to move through our membrane. And they act sort of like tunnels. Again, they're just a protein, but their structure allows things like potassium ions to diffuse from high concentration inside our cell and move and diffuse out of our cell. So again, channels allow these ions to move in or out of the cell by diffusion. Otherwise, they can't get through the phospholipid bilayer, charged ions like sodium, chloride, and potassium. They can't get through the phospholipid. So the way they can get through are through these channel proteins. And so potassium goes through one type of channel. Sodium is able to go through another set of channels based on its shape. Again, it's always from diffusion from high to low. So in this case, sodium moves into the cells, and they carry positive charge, and if you've ever heard of an electrical impulse, this is usually sodium rushing inside your little cells in your body. Some of our ion channels can be always open, and sometimes they can be closed or gated, so they can only open sometimes. So, for example, here we show an open potassium channel. We then show the sodium channel opening up. Sodium rushes in. We call those gated channels, meaning they open and close whenever your cell needs them to open and close. So remember, when sodium rushes in, it will diffuse into the cell. And so for neurons and heart cells, when sodium rushes in, those little cells get excited. And then when they rest, those channels can close. Chloride has channels as well. Those are important in the brain, for example. If chloride moves into your brain cell, it sort of shuts them off, turns them off. And so there's drugs that can actually target and open those chloride channels, maybe shut down parts of your brain and not make you so anxious. Okay, another important type of membrane protein, we call those transporters. So channels, you can think of channels like little tunnels. So some things are too big to get through membrane channels. Something like glucose, for example, it's too big and it can't actually get through a channel. So instead, we've developed, or cells have developed, transporter proteins that can grab the little glucose and, depending on its direction of diffusion, move it into or out of the cell. Since most of the time glucose is high on the outside of our cells, say after you eat a Snicker bar, glucose is floating around outside your cells, that little transporter protein for glucose will grab the glucose, bind it, and shuttle it through the phospholipid bilayer. So again, it's a little different from a channel, and glucose wouldn't otherwise be able to get into the cell. Examples in the kidneys and intestines, we actually have protein transporters that can actually grab a potassium, a sodium, and a chloride and shuttle them all through into the cell. So sometimes ions are actually grabbed by these transporters, and usually it always relies on diffusion. Sometimes, though, the cells will want to move ions against diffusion. So what if a cell wants to move sodium out and potassium into the cell? Again, that's against the direction of the diffusion of those natural molecules. So if you want to grab a potassium molecule and move it in, you're going to need to use a special transporter protein. And in our cells, we have transporter proteins that can grab potassium, move them into the cell, grab sodium ions and move them out. But because it's working against diffusion, it requires ATP. We call these pumps. So I want you to remember that. Pumps move ions against a gradient, against diffusion, and it requires ATP cache from your cell. So sometimes they're called ATPase pumps. Okay, something to remember is that sodium's high on the outside of our cells, potassium's high on the inside of our cells, and so you can see which direction they'll naturally diffuse. Pumps are actually able to transport ions against that diffusion gradient. So that's what makes it a pump transporter rather than a channel or just a regular diffusion transporter. What happens if those pumps stop working? It's important because cells start to swell and die. So it's important that those pumps always work in your cells. Some of the most famous membrane proteins are called receptors, and these are most famous often because they bind to hormones and they also bind to drugs, or pharmaceutical drugs that you might take. So, for example, if we take a heart cell in our body, it can actually, and we call those myocytes, they're going to have receptor proteins, membrane proteins that we call receptors, that can bind specific chemicals. For example, epinephrine, you might know it as adrenaline, is able to bind to a very specific protein in the membrane, and you might want to call it the epinephrine receptor, but because it binds several other chemicals, it's actually called the beta receptor for short. Beta adrenergic receptor, you don't have to memorize that, but you can remember the beta receptor. Okay, so what happens when epinephrine binds to a beta receptor? It tells your heart to pump faster and harder. So again, just remember what adrenaline does. It does that by binding to a membrane protein. And it binds and releases epinephrine depending on how much epinephrine is around. And then the little heart gets a signal to either increase its rate or decrease its rate. Okay, so you may have heard of a drug called a beta blocker. Beta blockers it's a drug that actually blocks physically the ability of epinephrine to bind to the beta receptor. So we call it a beta blocker. It decreases heart rate and the force of contraction, which then decreases your blood pressure. So that's a really important and common blood pressure drug. Again, the reason I talk about receptors is because they're often drug targets, and they're often targets for chemicals that our cells use to communicate. An example in your brain cells called neurons, there's actually receptors which bind a chemical called opioids. And you might know opioids as, at least synthetic opioids, as Vicodin and OxyContin. These drugs act like your own body's opioids and can bind to the opioid receptor and change the activity of your neurons, and it actually shuts off their activity, which then blocks pain sensation and pain signals in the brain and the spinal cord. So again, your body makes these chemicals. You can take drugs that also activate this receptor in your membrane, and these chemicals then turn off pain. I noticed I was spelling opioids wrong there, so that's the correct spelling. Okay, the next type of membrane protein that we're going to talk about are markers for the immune system. So again, we're just still talking about proteins in the membrane. So say you have a red blood cell, and these red blood cells have little markers on their surface. Some of them, if you know your blood type, are carbohydrates like the A and the B marker, but there's one called the RH marker that tells you whether you're positive or negative. It's actually a protein. So if you make the marker, you're positive. If you don't make it, you're negative. And we make lots of other markers that some are proteins, some are carbohydrates on our cell surface. The final category of proteins in the membrane and connected to the membrane I wanted to talk about are structural support proteins. And so our cells are not typically round little balls, but instead fancy shapes like erythrocytes, which are discs, neurons, which are very complex. And so one of the ways you make the shape of your cell complex is you use anchoring proteins in the membrane, and you anchor them to cytoskeleton proteins, which we'll learn more about. But this gives your cell its shape by anchoring the membrane to this network of proteins inside your cell. Kind of like a scaffold, you can make cells discs or star-shaped. Again, it requires proteins. And then the other thing are these these membrane proteins can also anchor cells to each other. So again, your cell proteins that are in the membrane can link to other cells. And so, for example, keratinocytes in your skin. All right, we'll talk more about cytoskeleton proteins inside your cell in a second. Explain how substances can move into and out of cells. And you might be thinking, well, wait a second, we were just talking about that. Yes, channels, transporters, including the pumps and the diffusion transporters, will help transport things across our cell that can't make it across the phospholipid bilayer. So again, to summarize, some things can't cross into the cell without help using proteins. Some things that are small and not charged can actually get directly through the phospholipid bilayer. So we'll look at some examples in a second here. But it's good that the cell can control most of what gets in and out using these proteins in the membrane. So an example of things that use proteins to get in and out: sodium, potassium, chloride, even water needs a protein to actually get through the membrane. And then other things can actually get straight through the phospholipid bilayer. Most of these are easy to remember because they're the gases. They diffuse straight through, like oxygen and carbon dioxide. Some hormones that are lipid friendly, like estrogen and testosterone, can also go directly through the phospholipid bilayer. Alcohol is another example. Sometimes your cells like to get big stuff in and out. And there are two processes, one called exocytosis, which moves stuff out, and endocytosis, which moves stuff in. And again, we're talking about bigger stuff here. And there's also a specialized sort of version of endocytosis called phagocytosis. So again, what does this mean? Well, endocytosis can move stuff into the cell or even pull things out of the membrane, such as a protein. Exocytosis will either release things out of the cell or in some cases add proteins to the surface of the cell, like a receptor or a new channel. So again, endocytosis, exocytosis are sort of bigger stuff targeted into or out of the cell. Cells have specialized structures called organelles, and these specialized structures and often compartments have certain functions within our cell. And we're going to focus more on this in lab next week, but we can also learn a little bit as an introduction right now. So if you quick pause this, you can identify all the organelles either in the textbook or in this cartoon version. and this is also available probably in your textbook or Wikipedia. So we want to be able to identify the organelles and also know their function. So one of the most famous organelles is this compartment in the middle of a cell called the nucleus. Again, whenever we show compartments inside our cell, when these organelles are compartmentalized, it uses the same phospholipid bilayer, just like the membrane of the cell. Okay, so the nucleus is famous for containing our genetic information called DNA, and sometimes the DNA is organized into chromosomes. So those things would be found in our nucleus. There's also a compartment found around the nucleus called the endoplasmic reticulum. I like to think of it as a factory network because it builds stuff. So it may build lipids, it may build carbohydrates that the cell needs. Also floating around in our cell are structures called ribosomes. These ribosomes are little protein builders. Some of the ribosomes float free in the fluid of the cell, and others are stuck on the surface of the endoplasmic reticulum. So we notice on a microscope that some of the endoplasmic reticulum is smooth without ribosomes, and some of the endoplasmic reticulum is rough because it has ribosomes. Again, ribosomes just make proteins. Okay, we also have these little structures for transportation around our cell called vesicles. So if you want to send something from the ER elsewhere in the cell, you'll use these little cargo containers called vesicles. They're basically little round spheres of membrane. And often the ER will ship its contents to this thing called the Golgi complex, which is usually located between the ER and the membrane. And so in this case, the vesicles fuse with the Golgi complex compartment, and then in there we have shipping and packaging, and then eventually sending those little vesicles out from the Golgi, either inside the cell where they can do their job, or often they then send the vesicles which fuse with the membrane of the cell. And so the contents are either dumped out of the cell like a hormone, or in some cases you'll add those receptors or proteins that you're building to the cell membrane. Another famous organelle is the mitochondria. Plural is mitochondria, singular is mitochondrion. Those are famous for making ATP, which we're going to talk lots about. You need nutrients and oxygen for those mitochondria to be able to make ATP, which is sort of like cache energy for the cell. If you ever need more ATP, you'll probably need more mitochondria. I wanted to mention that smooth ER has sort of a famous function, and that's to detoxify chemicals in your liver and your kidney. We have some other large vesicles that you might see in the typical cell. Lysosomes digest big material, so they're sometimes called the garbage disposal. Peroxisomes, they actually are chemical processing vesicles, and so they can detoxify things like hydrogen peroxide. So again, these are some things you'll see in the typical cell. Another thing we already talked about is how do you give the cell its shape and organization. That's called the cytoskeleton or cell skeleton, and those are proteins. Some of them are big, thick proteins, some of them are smaller, thinner proteins, but they're involved in organization of the cell, its shape. Sometimes trafficking, those little vesicles will walk along these big, long cytoskeleton proteins. When we learn about muscle cells, these proteins allow the cell to contract. We often see these, not often, but on certain specialized cells, we see these things called cilia. And they're these big projections which have cytoskeleton proteins inside of them that can help the cell move stuff like mucus. And if you smoke cigarettes, your little cilia will stop working. Other projections we see in some cells are called microvilli. I like to think of them as little ruffles in the cell membrane. It increases the surface area of the cell membrane so that you can move more stuff in and out of the cell. So it's good for transport because you have more space. So for example, if you're a stomach cell and you're pumping out acid, it gives you more space to pump out that acid. So microvilli are most famous in our digestive tract, including the stomach and intestines. Let's go back and look at more about ATP. So remember, ATP, I like to think of it as cash in our cell. If you don't have enough cash or ATP, your little cells can get sick and sometimes die. So we always need to make enough ATP or chemical energy to keep our cells being able to do the useful things that cells do. It's not really the money or the ATP, but what the cell can actually do with that ATP that's important. things can it do? It can pump ions around, it can contract, it can do chemical reactions, and it can process things if it has enough ATP. Most of the ATP, but not all of it, is made by mitochondria. So let's look at how our cells make ATP. If your cell has a glucose molecule, through several chemical reactions it becomes pyruvate, and in the process we make some ATP in our cell. And that's pretty cool because it doesn't require any oxygen. We call this process glycolysis. Then that pyruvate usually will go into our mitochondria, especially if there's enough oxygen around. If there's not enough oxygen around, then you'll make lactate or lactic acid from the pyruvate. But again, in our normal cells, that goes into our mitochondria, we mix in some oxygen and we make tons of ATP. Along with that ATP, we might make some CO2 and heat as a byproduct of that ATP production. So that's how you go from glucose all the way to ATP. We also, our cells like to use fatty acids and in some cases amino acids to also make ATP in our mitochondria as well. Again, you need oxygen for this to occur. Some cells and some bacteria don't need any oxygen. They only use glycolysis to make ATP, but most of the cells in our body need oxygen. We call that oxidative phosphorylation or aerobic phosphorylation when we produce ATP in our little mitochondria. So imagine if you go from rest to exercise, you might notice that you breathe really heavy. Why is that? Well, when you're exercising, your heart muscles, your brain muscles, your skeletal muscle cells are going and very active, and so they require lots of oxygen. They produce lots of CO2. So at the cellular level, you require more oxygen, and you're making more CO2. So in terms of your body, you have to breathe really heavy to help maintain oxygen and get rid of CO2. So chemically, when ATP is used up, your little cells actually break off one of those phosphates. Usually a protein will do that. And in that instant, it uses the ATP. It uses the cash to contract or move or do some sort of chemical reaction. And so again, if you don't have ATP, your little cells will be unhappy. Okay, the final learning objective is a bunch of vocabulary, and some of it you probably already can figure out, so not too big of a deal. When cells divide, you make one cell becomes two cells become four cells. We call that cell division, and you've probably heard of that before. Sometimes we'll call it proliferation. Cell proliferation means you go from having one cell to having many cells. So an important part of cell proliferation and cell division is called mitosis. And mitosis is really specifically the doubling of the genetic material and creating two nuclei, a nucleus for each of the new cells you're going to make. So the cool thing about cell division and mitosis is we get two identical cells and all the same genetic material. When cells get sort of old and sick, they undergo cell death, which we call apoptosis. So again, stem cells and cells that can regrow body's tissues are really cool because they can undergo cell division or cell proliferation and make new cells. And so those cells then mature and become what we call specialized. When they become specialized, they might become a skin cell or a blood cell, something specific and do a specific job. Stem cells, on the other hand, are called unspecialized because they don't quite have a specific job yet. Again, when cells get old, they usually die by apoptosis. Sometimes those cells don't die the way they should, and they can become tumor cells or cancerous cells, which we'll learn about in a lecture later. So again, these are just some vocabulary terms I wanted you to feel comfortable with. If you ever hear of progenitor cells, they're usually talking about stem cells. We have those in our adult bodies so that we regrow new tissues all the time, right? You grow new skin cells and new blood cells and new bone cells all the time, rebuilding your body. The downside of this is sometimes our cells grow out of control and we get something called cancer. Okay, that's the end. I'll see you guys in class. See ya.